Hi everybody, my name is Dragan. I am a third-year Ph.D. candidate in the Department of Biological Chemistry, and today I will be presenting to you my poster on understanding your skin as barrier tissue, especially in the context of these two transcription factors or proteins, OVL1 and OVL2. And so why am I interested in studying the skin? Well, the skin is our very first line of defense against external aggressors, both physical and pathogenic. And if we want to understand how the skin works, we need to understand what it looks like. So on the left image here, this is a cross-section of human skin. And when we're discussing skin barrier, what we're really talking about are those top two layers, the tan and dark pink colors called the epidermis. And so if I zoom in on the epidermis, in normal skin, you can see that the cells within this tissue are very tightly close to one another. However, if you get disrupted skin barrier, these cells are no longer close to one another. And so when this happens, you can actually get inflammation to occur. And so cases where barrier defects are linked with skin inflammation are psoriasis and atopic dermatitis. And what's interesting here is that if you get prolonged inflammation, this can actually transfer to other tissues. So in this case, here's one example where eczema or atopic dermatitis can eventually progress throughout the body, and it can eventually lead to, like, let's say, for example, asthma within the lungs. So one of the major things that my project focuses on is understanding how exactly the skin can communicate with these other tissues, such as like the lungs, or in my case, fat pads. And so, again, as I mentioned before, I'm interested in understanding this in the context of these two proteins and their regulatory proteins called OVL1 and OVL2. And OVL1 is important for, in other words, like the maturation of your skin cells, whereas OVL2 is important for kind of like the growth and development of your skin cells. And what our lab is currently showing is that when you lose OVL1 in the skin, this actually creates a susceptibility to inflammation. And in atopic dermatitis, OVL1 is actually a risk locus for skin disease. And so, as I mentioned before, when you lose OVL1, you can actually get a worsened inflammation or exacerbated inflammation. And so in this case, you can see that the mice have worse psoriasis-like condition, worsened barrier defects, and subsequently worse inflammation. And so one major question, again, I have is, what exactly do OVL1 and OVL2 do in adult skin? And what exactly is their role in terms of maintaining our skin barrier function? And so in order to understand both of these proteins, we've actually deleted both of them in the skin specifically. And when we do this, we can actually see that first barrier defects develop, then excessive nail growth, followed by a separation in body weight. And if you age these mice, all of these symptoms become worse. And so, as I mentioned, here you can see in the graph that these mice have worsened barrier defects. And if you take zoom in images of the skin of these mice, you can tell that the cells are not adhered to one another quite so well. And here is an example of the nails actually being longer in the knockout compared to the control. And so also the next thing that develops is weight loss. And so in younger mice, you can see that they already are losing weight, and as they age, it becomes worse, despite the fact that these mice are moving less and actually eating more, which is contrary to what you would expect. And this weight loss seems to be specific to fat tissues. And so even within the dermal, the fats within the dermis, the skin rather, you can see a reduction in fat content. And so what's interesting here is these mice appear to be burning more carbohydrate stores compared to the control mice. And so overall, what I think is happening is that when you lose OVL1 and OVL2, this causes skin barrier defects. And because of this barrier defects, these mice are now having total body defects or metabolic defects throughout the body. So I'd like to thank my lab and the ARCS funding for all of this research, and thank you so much for your time.
Hi, my name is Gianna, and I'm an MD Ph.D. student in Joan Steffen and Leslie Thompson's lab, and I'm a second-year ARCS scholar. Today I'm going to talk about how ApoE may be degraded by a process called autophagy. ApoE is a lipid-carrying protein, and the predominant lipid-carrying protein in your central nervous system, or your brain. Humans have several different types of ApoE, including ApoE4. ApoE4 is the biggest genetic risk factor for late-onset Alzheimer's disease. There are many hypotheses as to how ApoE4 causes such a great risk. One of them is that ApoE4 levels are lower than the other types. This may be due to increased degradation, although no one knows exactly how ApoE is degraded. Other secreted proteins have been shown to be degraded in an acidic organelle called a lysosome, through a process called autophagy. I hypothesize that similarly, ApoE might also be degraded by autophagy. There are several types of autophagy. One is macroautophagy, in which proteins get wrapped up by a membrane, and then that membrane fuses with the lysosome. Another type is called microautophagy, in which the cytosolic proteins enter the lysosome directly. I first tested whether ApoE might be involved in a type of microautophagy requiring a protein called LAMP2A, which has previously been shown to be required for cholesterol trafficking. I knocked down LAMP2A and found that ApoE accumulated in cells, suggesting that its degradation was blocked. I performed a similar experiment knocking down a protein called ATG7, which is required for macroautophagy. And surprisingly, I didn't see any change in ApoE levels. This suggested that ApoE degradation does not require macroautophagy. However, I also found that when I knocked down ATG7 or another protein, Rubicon, that is required for endocytosis involving autophagy proteins, that ApoE didn't enter cells from the media as effectively. So here I've taken media conditioned with fluorescent ApoE and put it on cells that are not fluorescent. And over time, we can see that fluorescence increases in control cells. However, in ATG7 or Rubicon knockdown cells, there is much less fluorescence, suggesting that ApoE is not able to enter the cell from the outside through endocytosis. In conclusion, we've found that ApoE is degraded by an autophagy process requiring LAMP2A. A future direction that I'm really interested in pursuing is whether this can also happen in animals outside of cell culture, and I've forged a collaboration with a lab at Harvard to try to answer that question. Another finding is that ApoE endocytosis seems to require some autophagy proteins, like ATG7 and Rubicon. I'd like to thank my PIs, Joan Steffen and Leslie Thompson, my undergraduate, Nikki Geller, the entire Thompson lab, and my many collaborators, including members of the Blurton Jones lab, fellow ARCS scholar Amanda McQuaid, and collaborating labs at other institutions, including Harvard and in Canada as well. ARCS funding has really helped me sustain these collaborations even during the pandemic shutdown, and that's something that I'm very grateful for. ARCS has also provided me with the support that I need to purchase electronic materials to analyze data, software to analyze data, as well as travel expenses for collaborations and also for conferences. So I'm incredibly thankful for your support, as well as the support of my other funding sources and my departments. Thank you.
Hello, my name is Bre Hoover, and I am a fifth-year PhD student in Angela Fleischman's lab, and I'm excited to share what I've been doing for the past few years. So every day, our bodies produce 200 billion blood cells in a precise balance of blood cell types. The blood-forming stem cells that reside in our bone marrow are the cell types that are in charge of keeping this balance. However, over time, these blood-forming stem cells will acquire a mutation that causes blood cancers. So in our lab, we study a particular type of chronic leukemia called MPN, that is caused by the acquisition of this mutation, JAK2V617F, in the blood-forming stem cells. And this drives blood production out of balance and produces too many red blood cells or platelets. And we have a mouse model to study this disease. But what's interesting about when introducing this mutation into mice is that it significantly reduces their lifespan to 3 months of age, where the typical lifespan for a mouse is 2 years. So it's been presumed that this severe drop in lifespan from this mutation is due to a thrombotic event or a blood clot, similar to deep vein thrombosis or other method. What was striking was that when I treated mice with an antioxidant drug called N-acetylcysteine, or NAC, this significantly improved their lifespan. While the standard treatment of care, aspirin for MPN patients, did not improve survival in these mice. Unfortunately, I don't have time to show the data, but you can look for yourself in my paper in Blood Advances from earlier this year, where we found that an interaction between these two mature cell types is what's key for the thrombotic event in these mice. So using a method called flow cytometry, we found that NAC has no effect on platelet activation, and platelets are the cell types that contribute to the blood clotting. However, NAC is blocking this aggregation between platelets and a particular type of white blood cell type called neutrophils. So there's a relatively new phenomena in the neutrophil field called extracellular traps, where neutrophils will expel out their DNA, typically to trap pathogens. But in aberrant situations, like in NPN patients, these neutrophil extracellular traps can contribute to thrombosis. And so this is very exciting data, and this work provides a preclinical rationale for investigating NAC as a clinical trial for MPN patients at UCI. And that's some work that my PI, Angela Fleischman, is working on. So I'd like to thank all the people that helped contribute to this work. I'd especially like to thank Barbara Hemkalo family and the rest of the ARC ladies. Due to your support, I was able to share my work at multiple international conferences around the country, including in Atlanta and Orlando, and I was able to have multiple resources to thrive in grad school. So I'd like to thank you for your time and attention.